buenos días a todos. Bienvenidos a este webinar que ofrece Derquimica junto a ABR Research. Es una empresa que utiliza una nueva tecnología para obtener principios activos botánicos a través de la replicación celular. ELIODORA DAL MONTE, responsable de compras en ABR, junto con ANDREA CARPI, director científico, nos van a explicar con más detalle este proceso llamado cariogénesis, así como las ventajas de utilizar toda esta nueva tecnología. Si durante el webinar surge cualquier duda o pregunta, las podéis dejar por escrito en el chat y tras la intervención de los ponentes pues podremos intentar resolverlas o incluso podemos daros acceso y hacer la pregunta en vivo. Si durante los próximos días también hay dudas, nos las podéis mandar por email, nosotros las transmitiremos a ellos mismos y este webinar va a quedar grabado, o sea que si en cualquier momento queréis recuperar el vídeo y revisarlo, pues no habrá ningún problema. Sin más, damos la bienvenida a ELIODORA DAL MONTE y Andrea Carpi. ELIODORA, como habéis visto, habla español, pero se siente más cómoda haciendo la presentación en inglés, pero si luego tenéis cualquier pregunta, pues en español ella las puede resolver perfectamente. O sea que sin más, chao ELIODORA, un piacere y empezamos.
Hola, hola a todos. So I will speak in English, but please don't hesitate if you have the question also in Spanish. I can also speak Spanish. So first of all, yes, I'm on the sales department, and there's also with us for more technical questions, also Andrea Carpi, our scientific director. Now I will share with you the presentation. Sorry, one second. Okay. Okay. On the presentation mode. Okay, can you see my slides? Okay.
Sí, ELIODORA, ningún problema, adelante.
Okay, so our company, ABR Research, is a company based in Italy. Our mission is to preserve natural resources by means of biotechnologies. So our big know-how, it's our technology that we're going to talk about it today, that it's the cell culture. Plant cell culture. So our vision is that we are engaged with nature and to entrust the biotechnology to develop top quality ingredients to support the human health. So what are the key facts of our technology? It's the environment preservation, the nature plus the biotechnology application, the standardization of the process, the purity, and our expertise in how to do that. So ABR, it's for active botanical research. We are in the business in different fields. We are in the health with the human, pet, and zootechnics. Human, most of our business it's in supplement, so for food supplements. Then we are also in cosmetic and food, like functional food. As well, this technology can also be applied in pharma and other things. And what we work also a lot, it's with some project customized for our client. So for all the fields, we can create for you, for other people, a proprietary cell line. So inactive and metabolites that a company is looking for. We can develop it and do the work for them and be their manufacturer. So let's go more in details on what is the plant cell cultures. So these plant cell cultures can be developed for the metabolite production. For which application? Pharmaceutics. Then you will see, along with the presentation, an historic view of this technology and also some examples that are on the pharmaceutics side. then nutraceuticals, nutricosmetics, functional food, medical advice, and cosmetics. So a plant, usually, everybody know the extracts. So the extracts are coming from plants that they grow in fields. The plants then are brought together, cooked or fermented or do whatever to get to the point, to the extraction, to arrive to the secondary metabolites. So these plant cells are the primary source of the production of any active botanical. So our technology, what do we do here? So first of all, we get a selection of plant species. So we are choosing which plant we would like to use. We go and pick it up from botanical gardens. So the most perfect plant that we can find. So botanical gardens, university, or around the world, some also rare plants that are the most interesting. Then what we do an initiation of an undifferentiated system. So we're starting to create from small leaves or from the seeds or from the flowers, but most of it from the seed. We're going to make the cell of the seeds grow. So we do the callogenesis with callus and then cell succession. This we do it with the clonal selection, genetic stability. We do the phytochemical profiling, omics analysis, and we optimize the process. So here you can see in these images, these are all the calluses that we can create from different plants. As you see, there are also different colors. We go and we create a field full of calluses. Those calluses are exactly the cells that contain the secondary metabolites. So when we arrive to this part that is the solid part of the technology, we have all these cells in petri dishes, so all agglomerated, that they produce the secondary metabolite. After that, there's a long study and a long work to select the most active. So the cells that are producing more secondary metabolite, the one that we are interested in. After that, from the solid side, then we continue to make the cells grow, and within the cells, their secondary metabolites. From the solid form, we go to a liquid form, and we go to a bioreactor for a large volume of production. So as you can see here, the plant cell culture is a technique then designed for the growth and multiplication of the cells and the tissue. What we use nutrient solutions that are based like on water and salt and some natural phytohormones. And they're all grown and multiplicate in an aseptic and controlled environment. So as you can see here in this image, on the left side, you have like the small leaves that we took. This first starting of the callogenesis goes in more solid callogenesis to go on to bigger container and to a big bioreactor in a liquid form. In the meantime, all these cells are continuing to grow. So what we do, after we arrive at this point, we scale up to bioreactors. This is the most difficult part for our company, and it's our biggest know-how, because we can really grow from small 1 liter liquid form to big bioreactor, 100 liters. And we do these big bioreactors all throughout a qualitative and quantitative control. We evaluate the biological activities. We control every time that the secondary metabolite is still there and still active. And we do the standardization of the substance active. We monitoring throughout the process for contamination, and we control the microbiological. So at this point, we arrive to a big bioreactor, more than one, but to explain you better, from one bioreactor, we just extract all the metabolites. We just crush the cells, and with an extraction based on ethanol, we extract the metabolite of our interest. After that, there's the downstreaming process, that is the formulation of cellular products. We do the packaging, the storaging, safety and quality control, and then order registration, certification, and distribution. So here it's a little detail of the process. As you can see, there's the target plant. We go to the cell culture stage with the selection of the higher producing cell lines, the one that are producing more metabolites. We go to a cell suspension stage to getting the best yields, to have the best results on the title of our product. Then we go to the bioreactor stage. We continue to monitor all the culture condition, the temperature, the agitation, the oxygen rate, and we continue to scale up with in bigger bioreactor. After that, as said before, we go to the downstreaming process and we arrive to the final product that is always checked with the title. I mean that at the end we arrive to a powder that has, for example, a 10% of actives. So this 10%, it's not in some lots 7, in some other production on batch 20. No, it's always 10. So it's really standardized. And what we check, we always check that we based all on our checking on the title of the product with the HPLC. So this is the description of the whole process. And here it's a summary of the process. As we said, taxonomical identification of the plant that we gather from the best botanical park and universities. We induce the calogenesis. We select the most active cells that are containing the most of the actives. And we go up to an industrial scale up of the cell cultivation in the bioreactor. Then there's the biomass water extraction and lyophilization. And we get to the botanical extract that we are looking for. And on the right side, you can see the final product packed. So now I will give you a little history of the technology when the plant cell culture were born. Andrea, maybe you can explain a little bit more about the history of the plant cell cultures.
Good morning. I want to take a little time to remind you some of the fundamental discoveries in the development of plant cell cultures. As you can see in this slide, the first essential date to remember is the 1902, the year in which Aberland demonstrated that plant cell can maintain totipotency. Cells derived from a specific plant tissue can potentially generate a new seedling. So the story begins like this. Over the next 30 years, hormones and vitamins that regulate plant growth and development were discovered. But the essential date is 1939, when Gottlieb, White, and Nobercourt established the first callus culture. This represents an essential moment in the development of plant cell culture. The potentiality of technology becomes evident. Over the next 20 years, cytokines were discovered, which in combination with hormones are necessary for cell growth and differentiation in plant and in vitro cultures. In the 1962, Murashin and Skog developed the first complete nutrient medium for plant and cell culture in vitro. But only 30 years later, Levin developed a bioreactor system for culturing plant cell and tissue. So this is a very important date. And in the 1990, finally, plant cell culture are used for production of plant metabolite and drugs. So this technology beginning to spread in the university in the world. And this is the overview of the plant cell culture technology. Now my colleague Eliodora continues the presentation.
Yes. So this is a little history of the plant cell culture, but to make you understand better how it started all and in which field it was applied, I will go with the success story. It was a drug that was produced with using plant cell culture. So the plant that was used is the Taxol. Taxol is a drug that is used to treat various types of cancer. It's one of the most famous anti-cancer in the world. But you have to understand that this Taxol, it's a plant that grows in thousands of years. So to get some percentage of the Taxol metabolite, you have to destroy a lot of forest and a lot of plants to get just a little. So when the Taxol was becoming really used in against cancer, there was a huge research on how to produce it better because the quantity was really little and the demand was huge. So between 92 and 94, a company that Phyton Inc. that was made from a couple of professors of the Ithaca University developed a plant cell fermentation technology in a large scale. So they started producing Taxol and the related taxane molecules without synthesis and harvesting the products from cultivated plants. So the same method that we're using in ABR as our technology was used at the beginning to create Taxol from plant cell culture. In 1995, Bristol Myers has invested more than 25 million to develop the industrial plant cell technology with Phyton. This company's fermentation facility used a 75,000-liter fermenter that they were capable of producing hundreds and hundreds of kilograms of Taxol in a single batch. So in 1998, the Taxol sales exceeded $1 billion. Most of it is produced by plant cell cultures. Today, the Bristol Myers company is the world leading manufacturer of Taxol, producing over 90% of the Taxol used worldwide each year. So this was an example of how our technology was applied in pharma. Now, let's go to our fields. So more like nutraceutic, nutracosmetic, cosmetics, pet, all the other sector that we're really interested in. So why now there's a big hype on plant cell cultures? Because there's a huge sustainability aspect. Nowadays, with the fossil fuels, emission, lost forest, there's a big challenge with the climate change. So, of course, everybody knows about that. Temperature will continue to rise. There are frost-free season. There are changes in precipitation. There could be sea level rise, more drought and heat waves. And Arctic will likely become more ice-free. Hurricanes more stronger and more intense. These make it difficult for regular extraction to have standardization, first of all, to have always the same price or more or less, because we are finding already in the market some actives that today they cost 10, tomorrow, next month, it's 100 because they're not available anymore. So Why? We call this technology net rise. Why? Because this symbol, it's a symbol of our extracts are totally environmentally friendly and biotech, so all controlled. And apart from the be bio, be pure, no pesticides, we are also saving, because right now the regular products are using soil that can be easily used for like foods that we all need with the growing population on the planet. There's water savings, no pesticides, because of course, when you buy supplements or extracts to put in your supplement, where you are getting extracts that are from fields, those fields, they're receiving rain. Most of the time, a lot of the time, there are acid rains. There are pesticides to make them grow better, fertilizer. And there could also be an impact from other nearby fields with GMO products. So our technology let us have a super pure product with soil saving, water saving, no pesticides, no fertilizer, and definitely GMO free. So what are the production advantages in our technology? We can preserve endangered species. Most of the clients, most of people that they come to us first, it's because they're looking for an active or a plant that is endangered, or it's already on the list of the rare plant on the planet. So they come to us because they have that claim, they want to keep that plant, but it's difficult through natural systems. So they come to us to create for them a line. So a cells that we can continue to make them grow to produce the secondary metabolites of their interest. Other, they come to us because they have a product on the seasonality. There are some period of the year where they don't have the products and they need the product. So our production is totally seasonality independent. There's no season for us. We just need the electricity, a little water compared to regular extraction, and our know-how. That is the biggest deal. Then we have a high degree of quality and safety of the product. We all, our company is all following the GMP standards. We are ISO compliant. So we have a high degree of quality and safety of the product. We also do all the technical analysis. We check for everything, even if in our technology, there's not the use of heavy metals, there's no pesticide, there's nothing. These we have the proof with the test and everything, but from where we start from just a little seed or a little leaf. Then it's the cells that continue to be monitored and constant controlling for the quality. Also, with our technology, there can be better planning and a more flexible production process for our client. So here, again, our platform, just some numbers on what is the difference between a regular extraction and a cell culture actives coming from cell cultures. So compared to field crops, we only use 0.0015% of the soils. The water that we use compared to the field crops, it's only 1%. The pesticide, I've already talked to you about that, and the fertilized as well. We only use water, some salt, and some sugars. And they're definitely not genetically modified. So right now we are almost at the end of our presentation. Of course, our regulatory, it's a little bit harder. Because even if it's the same secondary metabolites that is in nature, we produce it by the means of biotechnology. So for the new regulation, we have to do a novel food for what regards to nutraceutic markets. So we already have 3 products that are novel food approved. So they are already sold all around Europe and more, also US and Asia. Or some extent. These are the products that we have. We have Teupo. Also, one thing that I didn't mention before, in regular field crops, actually the title you can get, it's always maximum 10, 15%. With our production, we can really get near to the pharma level because, for example, Telpo, we have novel food approved for 10, 25, and 40. That is the title. So there are some titles that in nature don't exist. In our production, what we do, we always get to a title that is between 60 and 90%. So what we do to go to the market, we reduce the amount of the actives, adding simply maltodextrin, all certified and everything. So here we have the products that we have are Telpo, that is used for the inflammation bowel disease, Crohn's disease therapy. And it is the most used for to help to prevent prostatic hyperplasia. Then we have Actios, that is used as a nervous system. It's an anti-aging mind. It's a well state of oral gastric tract. It can treat hypertension, and there's a reduction of oxidative stress in smokers. So these claims that we're saying, they're all based on some research that were done to get to the novel food approved. And there's Equiplus, based on the echinacea, that is a sport supplement and a booster for immunity. We are also working on a lot of other products. Here it's a little pipeline for 2023. We have a product coming out that is ape liver, that is coming from a particular type of an apple. This product can treat or their adjuvant to the treatment of the dyslipidemia, hyperglycemia, liver steatosis, and liver fibrosis. Every time we start with a new product, we always do partnership and we work together with the universities or a company to check on the products, to do the test. For example, in the case of Up Liver, we are working with the Italian Liver Foundation, that we did already the test and everything to have these results. Then there's the ToxClean, that it's an adjuvant therapy for the Clostridium difficile infection disease. Then we are also creating a lot of new metabolites for cosmetics, most of it for anti-age, anti-inflammatory, and a lot more. So our point was really to make you understand what is this technology, the sustainability, and the strength of this technology. The rest, it's really open because we can do project on anything you are looking for. The only thing is that our processes are long. It's not that you give us a name and we do a cell culture in 1 week or a month. It's a process that can go from 1 year to 2 years. That's it. I think that is all for the presentation. I hope you understood exactly what is our know-how, what is our technology, and its sustainability. I give back to the other people for if they had the questions or if anything was not clear, please don't hesitate.
Muchas gracias, Eleodora. Gracias, mille, Andrea, también. Tenemos una pregunta en el chat. Nos comentan cuál es el precio comparado con otro tipo de extractos. Y evidentemente este tipo de tecnología, ¿no? Sube el precio en comparación de un extracto normal. Y si estáis trabajando también con otros extractos o lo hacéis bajo demanda. Y si los productos tienen algún tipo de estudio clínico.
Sí. I'm going to explain because I see the questions here. So the first question was, what's the price compared with standard botanical extracts? Actually, it's a little higher, but not that much, because if we would need to put the price of our research on the product, we would be totally out of the market. So right now it depends on which territory, but we are pretty much aligned with regular extract. There could be a little bit more, but it's really not that much. We always find we are already in the market, so we're selling a lot, our Telpo, Actios, And actually, they totally not out of the market. So it's not something like we already arrive at the point where we are not at the same price point, but pretty much there. Then, are you already working with other common extracts? Are they all on demand? No, we always have, like, our biggest part of the company, it's the research and development. So, of course, we are working with a lot of extracts. Sometimes we start ourselves and we go on. Sometimes we do some partnership at one point to get an exclusive product for somebody else. And sometimes we start from the basic on the client demand. But we already have a list of products that it's in the pipeline that we're still putting out on our own. Then all your products will be focused on clinical studies. So we always try, when we start our own, we always try, as I said before, to work with university, company, and do studies. Of course, at the point where we get to the novel food, we need the clinical studies. So of course we have it. But in most of the products that are still not novel food approved, sometimes is the client that want to take care of the clinical studies. So we give them the ingredients and they do the clinical study. Okay, then how are these ingredients legally named? Okay, so the name of the company, it's ABR. Then, as you saw in the presentation, there's this NetRise platform that it's a logo that we created to identify our sustainability, our platform of cell cultures. So this little logo can be put forward on the final supplements and can be used to raise awareness to the final client that they're using something that it's not normal, it's something better. Then the ingredients, there are some ingredients that are, yes, covered by a mark. Some others no, but it identifies already our products. And anyhow, when you have a novel food, you don't really need to legally name, because already the process of the novel food is like so time consuming and cost consuming. Then, ¿cuál es la cantidad máxima que se puede producir? Okay, so there's another thing that I just want to say a little, is that right now there's a hype on this technology. We know that there are other companies that they're doing some of that. We know them because they come to us for the quantities. So we were focused from the beginning on the nutraceutical and not cosmetic. Why? Because our biggest know-how and the difference with all the other competitors, it's that we have really high quantities. We can produce high quantity. If I remember correctly, but Andrea maybe will check on me, it should be 100 kilo per day. Andrea?
Yes, we have the possibility to produce about 2,000 kilograms of cell biomass for day, and we can extract about 3-4 kilograms of metabolites per day.
Okay.
Perfecto, pues creo que no hay ninguna otra más pregunta, Eliodora. Si queréis lo dejamos aquí, y en caso de que alguien tenga cualquier otra pregunta, pues la podemos hacer llegar tanto a Eliodora como a Andrea.
No hay problema, todo lo que necesitáis os dice.
Te agradecemos mucho, a Andrea también, y nos vemos pronto por aquí con el resto de gente. Un saludo para todos. Hasta luego.
Muchísimas gracias a todos.
Adiós.
Adiós.